So let's begin our today's topic. Last time we were discussing about polysaccharides. As I have told you, we classify polysaccharides into food storage polysaccharides, into structural polysaccharides. First, we classified them into homopolysaccharides and heteropolysaccharides. Now, I hope you understand what are food storage polysaccharides, what are structural polysaccharides. So, as I was telling you, structural polysaccharides, they are the polysaccharides which are involved in the formation of cell structures. So among structural polysaccharides, we discussed first cellulose and chitin. Those cellulose and chitin, they are included in homopolysaccharides. Why they are known as homopolysaccharides? Because their basic unit from which they were formed, that was similar, right? Cellulose is a polymer of monosaccharide, beta glucose. Similarly, chitin is a polymer of monosaccharide, modified sugar, N-acetyl glucosamine. So in chitin, many N-acetyl glucosamine units, they combine, they form the chitin. In cellulose, many beta sugars, they combine, they polymerize, and they form the cellulose. So in cellulose, the basic unit is beta glucose. In chitin, the basic unit is N-acetyl glucosamine. So that is why we call them as homopolysaccharides. Now we have got structural polysaccharides known as mucopolysaccharides. Today we are going to discuss about mucopolysaccharides. These mucopolysaccharides, they are slightly different from other polysaccharides. Other polysaccharides, starch, glycogen, cellulose, chitin, they were all polymers of monosaccharides. Other polysaccharides like starch, glycogen, cellulose, chitin, they were all polymers of monosaccharides. But these mucopolysaccharides, they are polymers. They are polymers of repeating disaccharide units. Disaccharide units. They are polymers of repeating disaccharide units. And each disaccharide is formed of two different sugars. For example, it may consist of one sugar known as hexuronic acid, and it may consist of another sugar that is known as N-acetyl hexosamine. So this is one sugar, this is another sugar. These two sugars, they combine together and they form a disaccharide. And many disaccharides combine together to form these mucopolysaccharides. So disaccharide plus another disaccharide plus similar another disaccharide, right? So many disaccharides, they combine. When many such disaccharides combine, they form the mucopolysaccharides. Now I hope you understand how mucopolysaccharides are different from other polysaccharides. Other polysaccharides like starch, glycogen, cellulose, and chitin, they are polysaccharides, but their basic unit is a monosaccharide, right? So they are the polymers of monosaccharides. But mucopolysaccharides, they are the polysaccharides, which are the polymers of repeating disaccharide units. They are the polymers of repeating disaccharide units. So here one disaccharide is first formed by combination of one sugar with another sugar. So this is a different sugar, this is a different sugar. So two different sugars, they combine, they form a disaccharide. Then many such disaccharides combine together and they form a long polysaccharide known as a mucopolysaccharide, right? So some of these sugars, hexuronic acid and acetyl hexosamine, some of them may be sulfated. Some of them may be sulfated. So they may also contain sulfur in them, right? So these disaccharides formed of hexuronic acid and N-acetyl hexosamine, they may be sometimes sulfated, right? Either hexuronic acid may be sulfated or sometimes N-acetyl hexosamine may be sulfated. Hex means 6, uronic acid. So it is a compound formed of 6 carbons. Hex means 6. So it is a 6 carbon containing sugar, right? N-acetyl hexosamine. It is a 6 carbon containing sugar having an acetyl group, right? So they are both modified sugars. So these two modified sugars, they combine to form a disaccharide. Some of these modified sugars may also have sulfur with them, right? So they form sulfated disaccharides. So one sulfated disaccharide, another sulfated disaccharide, these sulfated disaccharides, they may combine, they form polysaccharides known as mucopolysaccharides, right? Where we find these mucopolysaccharides? These mucopolysaccharides, they are structural polysaccharides. They are found in various body structures. That is why they are classified in structural polysaccharides. So I will show you an example of a mucopolysaccharide. I will show you an example of a mucopolysaccharide. How a mucopolysaccharide is formed. So let us take a hexuronic acid. Hex means 6. Let's take a hexuronic acid. So far it resembles with glucose. It is actually a glucose which is modified. It has got a COOH group here. In glucose, here we had on carbon 6, we had a CH2OH group. But in this modified sugar, CH2OH group is replaced by this HD group, COOH group, carboxyl group, right? So this is glucuronic acid. It is glucose which is modified, having HD group. So this is known as glucuronic acid. And generally we call such sugars, such glucose, which have got carboxylic group, carboxylic acid group, at their carbon first, at their carbon 6. This is carbon first, this is carbon second, this is carbon third, this is carbon 4, this is carbon 5, and this is carbon 6. At their carbon 6, they have got this carboxylic acid group, right? So when a glucose has got a carboxylic acid group at its carbon 6, we call it as glucuronic acid. So glucuronic acid is a hexuronic acid. Hex means 6. Uronic acid. So it is a 6 carbon containing group which has a acid at its carbon 6 position. So this glucuronic acid, it combines with another modified sugar. It combines with another modified sugar. It combines with another modified sugar. This modified sugar is N-acetyl. It is N-acetyl D-galactosamine. Galactosamine 6-sulfate. So it has got a sulfate group. As I told you earlier, some of these modified sugars may be sulfated. So it has got a sulfate group at carbon 6. This is carbon first, carbon second, carbon third, carbon 4, carbon 5, and carbon 6. So at carbon 6, it has got a sulfate group, right? So we call this substance as N-acetyl D-galactosamine 6-sulfate, right? So this is a hexuronic acid. This is an N-acetyl hexosamine. So they combine together by the formation of a bond. So one water molecule will be eliminated. A water molecule will be eliminated. A water molecule is here eliminated. This water molecule is lost. And a bond is formed between carbon first and carbon third. And a bond is formed between carbon first and carbon third. Are you able to hear me? Hello, are you hearing me? Okay. So bond is formed between carbon first and carbon third. So what will be this bond known as? One sugar combining with another sugar. When two sugars combine, what type of linkage they form? So this linkage will be known as, this is a beta sugar, this is a beta sugar. So it will be known as beta 1,3 glycosidic linkage. It will be known as beta. This is a beta sugar, beta sugar. The bond is formed between carbon first and carbon third. So this will be known as beta 1,3 glycosidic linkage. And this disaccharide, which is formed by joining up these two monosaccharides. This is one monosaccharide, this is another monosaccharide. So they combine, they formed a disaccharide. And this disaccharide is known as, chondroitin. Chondroitin 6 sulfate. It is known as chondroitin 6 sulfate, right? So one disaccharide is formed. Now one such chondroitin 6 disaccharide. One disaccharide plus another disaccharide plus another disaccharide plus another disaccharide, right? So many such disaccharides, they will form a mucopolysaccharide. Which mucopolysaccharide will be formed here? Which mucopolysaccharide is formed here? This mucopolysaccharide is known as chondroitin 6 sulfate mucopolysaccharide. So it is a polysaccharide of chondroitin 6 molecule, right? So these disaccharides, which are formed by combination of glucuronic acid and N-acetyl D-galactosamine 6 sulfate, known as chondroitin 6 sulfate when they combine together. Now many chondroitin 6 sulfate disaccharides, one disaccharide, another disaccharide, another disaccharide, another disaccharide, many disaccharides combine. They form this polysaccharide. And this polysaccharide is a polysaccharide of chondroitin 6 sulfate. Is it now clear to you how it is different from other polysaccharides? Other polysaccharides like starch, like glycogen, like cellulose, like chitin, they are polysaccharides of monosaccharides. But this polysaccharide, mucopolysaccharide, it is a polysaccharide formed of repeating disaccharide units. So first two modified sugars combine. One sugar here in this example is glucuronic acid. It combines with another modified sugar, N-acetyl D-galactosamine 6 sulfate. It has got sulfate group on carbon 6, right? So one monosaccharide combines with another monosaccharide and they form a disaccharide. What is this disaccharide formed? What is it is known as? It is known as chondroitin 6 sulfate. So first one disaccharide is formed. Then similarly another disaccharide will be formed. Another disaccharide will be formed. And many such disaccharides will combine and they will form mucopolysaccharides, right? So in this way many mucopolysaccharides are formed. Let us see what is the role of these mucopolysaccharides in our body. As I told you, they are classified in structural polysaccharides. Should I rub this? Okay. Well, many such mucopolysaccharides. For example, our first mucopolysaccharide was chondroitin 6 sulfate. It was our first mucopolysaccharide. Similarly, we have got hyaluronic acid. Hyaluronic acid. It is another mucopolysaccharide. Similarly, we have got heparin. Heparin is another mucopolysaccharide. Similarly, we have got keratin sulfate. Keratin sulfate. It is another mucopolysaccharide. So there are many mucopolysaccharides. I provided you the example of formation of chondroitin 6 sulfate, right? In the same way, hyaluronic acid may be formed, heparin may be formed, keratin sulfate may be formed, right? So what is the role of these mucopolysaccharides? This chondroitin 6 sulfate, it is an important constituent in the matrix of cartilage. It is found in the matrix of cartilages. It is an important constituent in the matrix of cartilages. These mucopolysaccharides, they mostly retain water with them. They are polar substances. They retain water with them. So they keep the tissues, body tissues, hydrated. They keep the body tissues hydrated. So it keeps the cartilage flexible and hydrated. So it is found in the matrix of cartilages. It is also found in the matrix of connective tissue. It is also found in matrix of connective tissue. Connective tissue. Right? It is also found in the matrix of connective tissue. Similarly, we have got hyaluronic acid. This hyaluronic acid, it is found in synovial fluid. Where synovial fluid is found? Synovial fluid, it is present in synovial joints. It is present in synovial joints. In this synovial fluid, we have got hyaluronic acid, and it acts as a lubricant. It acts as a lubricant in joints. It reduces the friction between two articulating bones at joints. What is a joint? Joint is a place where two bones articulate with each other and make some movements, right? The articulating place of two bones is known as a joint, right? So these joints where the two bones are making movement over each other, they should have a lubricant so that friction is reduced. So hyaluronic acid, it is a constituent of synovial fluid, which is found as a lubricant at the joints, right? So this hyaluronic acid, it also retains water. It also keeps this synovial fluid hydrated, right? So it is involved in the lubrication of the joints. Similarly, we have got heparin. Heparin acts as an anticoagulant. Anticoagulant in blood. Heparin acts as an anticoagulant in blood. It prevents internal coagulation of blood. And we have got keratin sulfate. This keratin sulfate, it is also found in connective tissue. It is also found in connective tissue. It also keeps the connective tissue hydrated. It keeps the tissue hydrated. It keeps the tissue moist, right? So these are some mucopolysaccharides which are found in various body tissues. So their main function is to retain water. They retain water in the body tissues. They keep the body tissues hydrated. And some of them also act as lubricants. For example, hyaluronic acid in synovial fluid. This hyaluronic acid also acts as a cementing substance between the cells. It keeps the cells adhered to each other. So it acts as a cementing substance between the adjacent cells. So it acts as a lubricant. It acts as a cementing substance to keep the cells of epithelium stuck to each other, adhere to each other, right? Then we have got heparin. This heparin acts as an anticoagulant in blood. It prevents internal coagulation of blood. And keratin sulfate is also found in connective tissue, which retains water in connective tissue, which keeps the connective tissue hydrated, right? So that is the function of mucopolysaccharides. So you have to keep it in mind that these mucopolysaccharides, they are structural polysaccharides, which slightly differ from other polysaccharides. Other polysaccharides like starch, glycogen, cellulose, and this chitin, they are polysaccharides of monosaccharides. These mucopolysaccharides, they are polysaccharides of repeating disaccharide units, some of which may be sulfated. So that is all about mucopolysaccharides, right? So with this, we come to the end of polysaccharides. Now, in cellular macromolecules, we are going to discuss about proteins. Now we will discuss about proteins. Protein. This word protein, it has been derived from a Greek word, proteos. Proteos. Proteos means something of prime importance. Something of prime importance. Something of prime importance. So the word protein, it has been derived from the Greek word proteos. The Greek word proteus means something of prime importance, something which is very important, which is of utmost importance, right? So these proteins, of what material they are formed of? Can you tell me? Proteins are formed of? These proteins, they are formed of amino acids. They are building blocks or amino acids. So we call amino acids as building blocks of protein. We say amino acids are building blocks of proteins. How many amino acids are involved in protein formation? There are 20 protein forming amino acids. Protein forming amino acids. There are 20 standard amino acids or protein forming amino acids, right? So these 20 amino acids, they act as the letters of English alphabet. How many letters are there in English alphabet? How many letters are there in English alphabet? In English alphabet, we have got 26 letters, right? From A to Z. From A to Z. Our whole English language consists of only 26 letters. Those letters are from A to Z. If you will count from A to Z, you will find 26 letters, right? But how many words are there in English language? Can you tell me how many words are there in English language? There are thousands of words, right? There may be thousands of words in English language. So those thousands of words of English language, they are basically formed from these 26 letters. When these 26 letters A to Z, when they are used in different combinations, they form different words. For example, if we use W, O, or D in combination, what does it form? It forms a word, right? If we will add just S in front of it, it forms another word, sword, but the meaning is different. Similarly, we have got 20 amino acids, as we have got 26 letters in English alphabet for formation of thousands of words. Similarly, we have got 20 amino acids. These 20 amino acids, by different combinations, they form thousands of proteins in our body. According to an estimate, there are some 10,000 different proteins. There are some 10,000 different proteins in our body. According to an estimate, there are some 10,000 different proteins in our body. So these 10,000 different proteins, they are formed. Please don't make noise. These 10,000 different proteins, they are formed simply from 20 amino acids by different combinations. Do you get me what I am trying to convey you? Hello, please don't make noise. Hello, please don't make noise. So as English language consists of thousands of words, those thousands of words are formed from 26 letters by their different combinations. Similarly, there are some 10,000 different proteins in our body. These 10,000 different proteins of our body, they are formed from 20 amino acids by different combinations, right? So when these 20 amino acids, when they get arranged in different combinations, they form some 10,000 different proteins in our body, right? So this protein, it exists in different structures. A protein exists in different structures. So we will see the hierarchy of protein structure. A protein is not found, it is not only having a single structure. It is existing in different structures. We then give rise to a different structure. So first we will have a look at structure of proteins. So how these proteins are formed, right? So there is a hierarchy in protein structure. First, we will have a look at hierarchy of protein structure. Protein structure. So hierarchy means arrangement in which smaller components of a structure are arranged first, then followed by a little more complex, then little more complex, then the most complex, right? So in this hierarchy of protein structure, first protein forms a simple structure that is known as a primary structure. So first there is primary structure. Primary structure. Then after primary structure, a protein may assume secondary structure. Secondary structure, right? Then secondary structure is followed by tertiary structure. Tertiary structure. And this tertiary structure is then followed by quaternary structure. Quaternary structure. So this arrangement of protein structure, first the protein becomes very simple. That is known as primary structure. Then this simple structure becomes a bit complex. That is known as secondary structure. Then this secondary structure becomes a bit more complex. That forms the tertiary structure. Then tertiary structure gives rise to little more complex structure known as quaternary structure, right? So when a protein is formed, first it will develop primary structure. Then it will develop secondary structure. Then it will develop tertiary structure. Then it will assume the quaternary structure. So this is known as hierarchy of protein structure. This is known as hierarchy of protein structure. So hierarchy means the arrangement, the system in which smaller components of a structure are arranged first, then followed by little more complex, then more complex and the most complex, right? So a protein does not exist in a single structure. A protein first forms a primary structure, then it forms a secondary structure, then it forms a tertiary structure, then it forms the quaternary structure. So we have to look at these structures one by one. We have to look at these structures one by one. So we will start with the primary structure. We will first start with the primary structure of protein. From how many amino acids a protein is formed? From how many amino acids a protein is formed? Come on, tell me. From how many amino acids a protein is formed? There are 20 standard amino acids which are involved in the formation of a protein, right? So a protein is formed just from 20 amino acids. So primary structure is the arrangement in which these amino acids are arranged. Primary structure is the arrangement in which amino acids are lying to form a linear polypeptide chain. Linear polypeptide chain. In primary structure, amino acids are arranged in a specific order to form a linear polypeptide chain, right? For example, there is one amino acid, there is another amino acid, there is another amino acid, another amino acid, another amino acid. So many amino acids, they are linked together. And when many amino acids get linked together, they form a polypeptide chain, right? This polypeptide chain, number first, it has got specific number of amino acids. Specific number of amino acids. In a polypeptide chain, the number of amino acid is specific, right? If we talk insulin, insulin has got two polypeptide chains. One polypeptide chain contains 22 amino acids. Another polypeptide chain contains 31 amino acids, right? So in every polypeptide chain, the number of amino acids is specific. So this is the first thing. Then second thing, that is sequence of amino acids. Sequence of amino acids in a polypeptide chain. In a polypeptide chain is also specific. Is also specific. Right? As I mentioned earlier, there the spelling of word is WORD word, right? It has got 4 letters. WOR and D. If I will change the sequence of these letters, will it remain the same word? If I will change the sequence of these letters, suppose I will write it like this, GROW. There are the same letters. There are the same letters, but they are in different sequence. So that changes the meaning. That changes the meaning. Similarly, in a polypeptide chain, the sequence of amino acids is always specific. If we have got a protein which is in its primary structure, it will have a fixed number of amino acids. That protein will first have a fixed number of amino acids. It will have a specific number of amino acids. Then those amino acids will be in a specific order. They will be in a specific order, right? If we will change the sequence of amino acids, then the protein will lose its function. The protein may be not the same, right? So every protein first forms a primary structure. Every protein first forms a primary structure. What is the primary structure of protein? In primary structure, the primary structure of protein is formed by the combination of a specific number of amino acids. The primary structure of protein is formed by combination of a specific number of amino acids in a specific sequence to form a linear polypeptide chain. So that gives rise to primary structure of protein, right? Are these proteins functional in primary structure? Are proteins functional in primary structure? Most of the proteins are not functional in primary structure. Most of the proteins are not functional in primary. Most of the proteins are not functional in primary structure. However, there is a protein known as insulin protein. Insulin protein. It has got only primary structure. Insulin protein is only having primary structure, and it is functional in this primary structure. As I told you, most of the proteins do not become functional in primary structure. Majority of the proteins are not functional in primary structure. They assume then secondary structure, then tertiary structure, then quaternary structure. But there is a protein known as insulin protein. This is the only protein which is functional in primary structure. So in primary structure, it has got 2 polypeptide chains. It has got 2 polypeptide chains. This is its one polypeptide chain, this is another polypeptide chain. In this polypeptide chain, in human insulin, there are 22 amino acids. In this polypeptide chain, in human beings, there are 31 amino acids, right? These 22 amino acids, they are in a specific order. They are in a specific order. These 31 amino acids, they are also in a specific order. So when these 2 polypeptide chains, when they get linked by disulfide bridges, when they get linked by disulfide bridges, this is a disulfide bond between them. Or we call it as a disulfide bridge. A disulfide bond is formed between two amino, A disulfide bond is formed between 2 sulfur-containing amino acids. Can you recall which are the sulfur-containing amino acids? There are only 2 amino acids which contain sulfur. Can you name them? It is methionine and cysteine. It is methionine and cysteine. Right? So insulin, it is formed of 2 polypeptide chains. Insulin is formed of 2 polypeptide chains, right? It is polypeptide A, it is polypeptide B, right? This polypeptide A, it is formed of 22 amino acids in a specific order. Polypeptide B, it is formed of 31 amino acids in a specific order. Then these 2 polypeptide chains are linked by disulfide bridges or disulfide bonds. Disulfide bonds are formed between sulfur containing amino acids and they get linked, and that makes this insulin, the functional protein, right? So in total, there are 53 amino acids in insulin. So insulin is the only protein which is functional in primary structure, right? Now, can you tell me what is the primary structure of protein? So a primary structure of protein is formed by a specific number of amino acids arranged in a specific order in a polypeptide chain, right? So here in insulin, there are two polypeptide chains, one having 22 amino acids, another having 31 amino acids. These two polypeptide chains having these 22 and 31 amino acids in a specific order get linked by disulfide bonds between their sulfur containing amino acids and they form a functional insulin protein. So insulin is the only protein which is functional in primary structure. I think we are running out of time. So this is sufficient for today. This is sufficient for today. We will discuss remaining of it tomorrow, inshallah. Any questions? Any doubts? कोई question, कोई सवाल? Please don't make noise. इससे disturbance होती है. Okay, thank you for being with me.
Thank you, sir.
Okay, thank you. I'm ending this meeting.